Welcome to Biology at Priyajit, my dear viewers. Today we explore one of the most fundamental biomolecules of our life, that is nucleic acids. Just as a book stores information and guides action, DNA and RNA as our biological macromolecules, they can store, transmit and express the genetic instructions of every living cell. From the double helical structure of DNA to the functional roles of different types of RNA in protein synthesis, nucleic acids control heredity, growth, and cellular activities. Let us understand biochemical structure and biological significance in a simple and systematic way. So the discovery of nucleic acid, it was discovered by Frederick Maser in 1869 from nuclei of past cells or leukocytes. He named it as nuclein. And later Richard Altman renamed it nucleic acid. Nucleic acids basically store and express genetic information. Types of nucleic acids, it can be DNA or RNA. They differ in type of sugar and one nitrogen base, that is thymine in DNA, and there is uracil in RNA. So nucleotide is basically the unit of nucleic acids, and each nucleotide consists of three components: nitrogenous bases, pentose sugar, and the phosphate group. So this is the two types of nucleic acid, RNA and DNA, and there are the nitrogen bases. Nitrogen bases are basically aromatic, heterocyclic, planar, weakly water soluble molecule, and they are derived from purine and the pyrimidine. These are the pyrimidine derivatives: cytosine, uracil, and thymine, and the purine derivatives are guanine and adenine. So purines are adenine, 6 amino purine; guanine are 6 oxy 2 amino purine; pyrimidines are the single ring structure; purines are the double ring structure. Pyrimidines are include cytosine, which is found in both DNA and RNA; thymine is found only in DNA, and uracil is found only in RNA. So thymine is absent in RNA and uracil is absent in DNA. Sugar component, there are two pentose sugars, ribose, which is found in RNA, and deoxyribose, which is found in DNA. So the differences: ribose has OH at 2' carbon position, and deoxyribose has H at 2' carbon position. Sugar puckering: sugar is non-planar and it shows puckering, envelope form or twisted form, and pucker type C2 endo, which is most common in DNA and purine, and C3 endo, which is common in RNA and the pyrimidine bases. So the phosphate groups, which are derived from the phosphoric acid, this is the phosphoric acid moiety, this is the phosphate group. They gives the nucleic acid acidic nature and negative charges. So this is the sugar component. Ribose has 2' OH and deoxyribose has 2' H. There is no oxygen, so it is named as 2' deoxyribose, as oxygen has been removed. Nucleoside, it consists of base and sugar. There is no phosphate. It is linked by N glycosidic bond. C1 of sugar is bonded to N9 of purine or N1 of the pyrimidine base. And examples include adenosine, guanosine, cytidine, uridine, and thymidine. So conformations are there as syn and anti-conformation. Pyrimidines are mostly found in anti-conformation, and purines are found in both syn and anti-conformation. So nucleotide consists of both the nucleoside and plus phosphate. The nucleoside consists of the nitrogen base and the sugar, and when phosphate group is attached to the nucleoside, it will form the nucleotide. This is the image. So nucleotides include monophosphate. There are phosphates at the 5 carbon attached to the 5 carbon. So types monophosphate, this is the alpha phosphate. When there are diphosphate, this is the alpha phosphate and beta phosphate, and when there are triphosphates, alpha, beta, and gamma phosphate. So polynucleotide chain, they are formed by the phosphodiester bond, and it is formed between the 3' OH of one nucleotide and 5' phosphate of next. So its direction is always 5' to 3', and two strands are always anti-parallel. That is complementary in nature. So this is the deoxyadenosine. It's a purine nucleoside, and deoxycytidine is a pyrimidine nucleoside. So this is the polynucleotide chain image. Nucleotides in DNA, these are the nucleotides in DNA, and these are the RNA nucleotides. These are the nucleotides in DNA. These are the nucleotide. There is phosphate, sugar, and nitrogen bases. Nitrogen bases can be two types: pyrimidines, cytosine, thymine, and uracil, and purines, adenine and guanine. So there are sugar can be two types: deoxyribose, which is found in DNA, and this is ribose. So when polynucleotide chains are formed, the nucleotides are bond with each other with the help of phosphodiester bond. Why it is named as phosphodiester bond? Because there are two ester bonds are formed. One ester bond bond is formed within a nucleotide between a phosphate and a sugar, and the second phosphate group is formed between the phosphate group and with the next nucleotide sugar. So there is a bond. This is known as phosphodiester bond. It is responsible for formation of the polynucleotide chain. So this is RNA nucleotide and DNA nucleotide. This is the RNA nucleotide, DNA nucleotide. This is the differences between DNA and RNA. DNA is a double-stranded structure. Sugar phosphate backbone is there. RNA is single-stranded. It is also a sugar phosphate backbone, so it has ribose sugar, but the DNA has deoxyribose sugar. DNA has thymine and RNA has uracil. But cytosine, guanine, and adenine bases are found in both DNA and RNA. So sugar structure of DNA, that is bDNA, that is Watson-Crick model. It is basically a right-handed double helix. So sugar phosphate backbone is present outside. This is the sugar phosphate backbone. These bases are found inside the sugar phosphate backbone, and they are paired as A double bond T, that is 2 hydrogen bonds, and G triple bond C, that is 3 hydrogen bonds. So features include 10 base pairs per turn, and distance between base pairs are 3.4 angstrom, and one turn there this is 34 angstrom. So there are major and minor groups are present. C2 endosugar is endopuckering is found. So according to Chargaff's rule, the A or adenine will be bonded with T and G will be bonded with C. So there are equal number of purine and pyrimidine. So AT base pairs are found, and there is GC base pair, GC interaction. There are triple hydrogen bonds and there are double hydrogen bonds. So you can find here the formation of the hydrogen bonds between adenine and thymine and guanine and cytosine. How they make the bonds, you have to follow through the structures. So there are different forms of DNA which vary among each other. There are aDNA, bDNA, and zDNA. aDNA, it is right helix. bDNA is also right helix, but zDNA is left-handed helix. In case of aDNA, there are 11 base pairs per turn, but in case of bDNA, there are 10 base pairs, or roughly 10.5 base pairs per turn, and in case of zDNA, there are 12 base pairs per turn. Sugar puckering, aDNA is C3 endopuckering, bDNA is C2 endopuckering, and zDNA is mixed type. Groove, it is a deep major group, and it is bDNA is standard, and zDNA is a zigzag backbone. Special feature, aDNA has a deep major group, bDNA has a standard group, and zDNA is a zigzag backbone. Special feature, aDNA is dehydrated DNA or anhydrous DNA, bDNA is normal DNA, and zDNA is GC-Duch, as they are zigzag in nature, and it has a regulatory function. So now we should go for the structure of RNA. Usually the RNA is single stranded. They contains ribose, phosphate, and nitrogen bases. There is A, G, C, and U. There is no thymine. So they are less stable than DNA because it has a 2' OH group, which is susceptible to the RNase enzyme. It has uracil instead of thymine, and they lacks histone protection. So types of mRNA, RNA. The first one is mRNA, which carries genetic information and acts as messenger RNA. It can be two types: monocystonic, found in case of eukaryotes, and polycystonic, found in case of prokaryotes. tRNA is an adapter molecule, brings amino acids. rRNA, it is a ribosome structure, 18S, 5.8S, and 28S and 5S. snRNA is small nuclear RNA, which is responsible for splicing. It forms the snRNA piece. snRNA, that is small nuclear RNA, which is small nuclear RNA is responsible for RNA modification. So gRNA is guide RNA, it is responsible for RNA editing, found in mitochondria. siRNA or microRNA, silencer RNA or microRNA, they are responsible for gene silencing. tmRNA, they rescue stalled ribosomes found in bacteria. This is the tRNA molecule. So hyperchromicity of DNA, hyperchromicity increases in UV absorbance. Hyper means higher, and chromicity means the absorbance of color. So when the absorbance increases in UV at 260 nanometer, when DNA gets denatured, that when double-stranded DNA is converted into single-stranded DNA. So it occurs due to breaking of the hydrogen bonds, unstacking of bases. So double-stranded DNA it's converted into single-stranded DNA. So absorbance increases by approximately 37%. And the TM or melting temperature at which 50% DNA is denatured. So high GC content, there is higher TM. Increase in salt concentration stabilizes the DNA and raises the TM or melting temperature. Hypochromicity decreases in absorbance during the renaturation process. So special DNA structures, there are non-bDNA structures. They include palindromic sequence, which form the hairpin or cruciform structures. When DNA has inverted repeats, the strands can fold back on itself, forming a hairpin or single-stranded and cross-shaped or cruciform double-stranded structure. Hook-strand base pairing, triplex DNA. A third DNA strand can bind to a normal DNA double helix using hook-strand bonds, forming a triple-stranded DNA or triplex. G quadruplex or four-stranded or G-rich DNA. G-rich DNA DNA sequences can form stable four-stranded structure held together by stacks of guanine bases. HDNA, they are polypurine or polypyrimidine mirror repeats. In sequences with mirror repeats of purine and pyrimidine, DNA can form a triple-stranded structure called HDNA. So, my dear students, we have now seen how nucleic acids, DNA, and RNA are the fundamental molecules of heredity and expression, from storing genetic information in DNA to transforming and translating it into proteins through various forms of RNA. They govern growth, development, and cellular function. So, as you prepare for university exams or competitive exams like CSIR-NET, remember that a clear understanding of their biochemical structure and biological significance is essential for mastering molecular biology and biochemistry. So, keep revising, stay curious, and continue exploring the language of life written in nucleotides. Thank you, and see you in the next class. Bye.